Thank you for standing by and welcome to the Arovella Therapeutics AACR poster webinar and Q&A today. All participants are in a listen-only mode. There will be an initial slide presentation lasting for approximately 30 to 35 minutes, followed by questions that have been received via email and others that come through live. So if you do have a live question you'd like to submit, please use the Q&A function within the Zoom app. On behalf of Arovella today, on the line we have the CEO and Managing Director, Dr. Michael Baker, and Senior VP of Development and Translational Medicine, Dr. Mini Bharathan. To begin, I'll hand it over to Michael. Please go ahead.
Great, thanks, Matt. We're delighted to be on the webinar today, talking about the data that we just presented at AACR, or the American Association for Cancer Research. And for those of you that don't know, it's one of the most significant cancer events on the calendar, certainly based in the US, in Orlando this year. And so we're delighted that our abstract was accepted and delighted to have presented. And we're going to talk through some of that data today, and hoping that people can take away a lot from it. But certainly delighted to have Mini on the call as well today, talking through some of that data. And we will be making forward-looking statements, so please do take note of our disclaimer. Now, as I said, we're looking forward to talking through the data, and I'd just like to start with some of the highlights up front and some of the key points that we will be talking through throughout the remainder of the webinar. And firstly, a key factor of cell therapies is that they can be well expanded, and we've demonstrated that with some of this data. That's important for making sure we've got enough cells for a lot of cells for patient doses. We've shown that ALA-101 kills cells that express CD19, including primary patient tumor cells, so actual tumor cells that come from patients, and using aggressive BALL mouse models, we've shown a significant extension of lifespan of those in those models with ALA-101. And following expansion, we've shown that the ALA-101, or the cells that comprise the product, have the ability to multiply further when they see the antigen or the target CD19, which is important for a therapeutic product. And also, once they have been stimulated, and this is what contributes to their ability to do what we want them to do inside of humans with cancer, is that they do produce anti-cancer cytokines. And we'll talk through all of those points in a little bit more detail. Okay, and so all of this combined, I think what it really points out, and with the significance, is that we've confirmed that our proposed manufacturing process maintains the effectiveness of cryopreserved or frozen ALA-101 when it's used off the shelf after thawing those out and taking them out of the freezer. Now, just a little bit of background. We did go through a webinar recently talking about iNK T cells, but I think it's just good to have a quick refresher. So invariant natural killer T cells are a naturally occurring immune cell. They've evolved to have what we call an invariant T cell receptor, which I'm highlighting here in pink, which recognizes something that's found on a number of different cancer types called CD1D. So they have already evolved to target and kill certain cancer types that produce that molecule. And what we know is because the ITCR, or invariant T cell receptor, doesn't change between people, and it always recognizes the same target, this is what enables us to take these cells from a healthy person and give them to a patient with cancer, because they don't cause graft versus host disease. So we also know that iNK T cells have numerous roles, and this is based on preclinical research, where they shape the tumor microenvironment by eliminating cells or blocking cells such as myeloid-derived suppressor cells, as well as tumor-associated macrophages. But they also play a role in potentially recruiting other components of the immune system in the host that may then join the attack on the cancer cells. So remember, when we engineer them with what we call a chimeric antigen receptor, or CAR for short, this is what gives us another turbocharge, and it's like putting a heat-seeking missile in the iNK T cells, that they can go and recognize a specific marker on the surface of those cancer cells. So when we have both the CAR and the ITCR engaged, our product becomes dual targeting. Now, just to step through this in a little bit more detail for ALA-101, what is it exactly? So this is our lead program or product, and it is a CD19 targeting CAR iNK T cell therapy. And so what that means is it's the iNK T cell that has a chimeric antigen receptor, or CAR, that targets CD19. Now, CD19 is a known target on cancer cells. It's on normal B cells, but on B-cell leukemias and lymphomas, and it's been validated as a suitable target through the approval of a range of T cell products. but we all know that those T cell products all rely on use of patient immune cells as the starting point. And so ALLO-101 is manufactured with what we call the 3rd generation lentiviral vector platform, which makes it up to the latest safety standards, and we've engineered it to contain genetic elements that have a proven safety profile in clinic and beyond, which we believe should set us up well for interactions with regulators such as the FDA. Now, in terms of our manufacturing scheme, this is what it looks like. We take the blood from our healthy donor. We then use sophisticated separation techniques to isolate the iNKT cells. Once we've separated them, we engineer them genetically to produce the chimeric antigen receptor, or CAR. They then get expanded into the billions before they're ultimately then put into vials, stored in the freezer. And so when we're actually ready to treat a patient with the therapy, we thaw those cells out, and then we can dose patients that are eligible to take that therapeutic. So why we think that's important is we believe that we're starting with healthy material, because we're not starting from a patient that already has cancer, so their immune cells may not be doing as well, and they haven't seen chemotherapy or any other types of therapies that may have damaged their immune system. We also know that because we're able to make multiple doses per batch, we won't have the same challenges that afflict the current T cell therapies, where you're making one product per patient, which is very, very difficult logistically, cost ineffective. We expect that enables to have us to reach more patients and have a more commercially viable product. Now, another big factor is to produce the cells on demand. Let's say for patients, it takes in the order of 4 to 6 weeks, and groups are working to bring that down, but there is a cycle time that you won't get below. And of course, all that means is for patients with aggressive diseases, there's the opportunity for that cancer to continue to spread and grow while that therapy is being produced, versus pulling it out of the freezer and then giving it to the patient that's eligible to take it. And of course, because the product's then sitting in a freezer ready for use, it does remove the risk of any manufacturing run failures. So I'd just like to summarize some of the advantages that I've just talked through, but in essence, we use mature iNKT cells from healthy adult donors. Now, this means we don't require any complicated reprogramming of stem cells, which is something that a number of other groups are working towards. We do see, when we're working with our lentivirus, high transduction efficiency rates of isolated iNKT cells, which is important because we want to maximize the number of iNKT cells that produce the chimeric antigen receptor. When we do that step, which is called transduction, where we deliver the CAR to the cells, this is performed on a very small number of cells in our case. And what that does is enables us to reduce the quantity of a relatively expensive agent, which again is good for the cost of goods for the product. Now, as I've mentioned, we have efficient expansion of the genetically modified cells, which is important again when we're trying to maximize the number of doses we could get from a single batch. And this is some of the work Mini's going to talk through today, but we've seen that the cells, once manufactured, maintain a very highly cytotoxic population, so they're quite effective at killing cancer cells. So with that, I'd like to hand over to my colleague Mini, and she will take us through some more of the data that we've seen that we presented at AACR this year.
Thanks, Michael. Can you hear me?
Perfect.
Yeah. So as Michael mentioned, here I'm showing you the data that we generated recently at Arovella, and that was presented at the AACR last Sunday. We isolated the iNKT cells from a healthy donor, as Michael explained before. This is specifically for the CAR19 iNKT cells, and then engineered with the lentivirus to express the CD19 CAR. And the cells were then expanded for 21 days and then were frozen down. And as you see from the graph presented on the slide, the y-axis represents the fold increase, and x-axis represents the days in culture. And as you can see from the graph, we were able to get 5,000-fold expansion of ALLO-101 by day 21. The significance is that the higher the fold expansion, the greater number of doses you could get from a single manufacturing run. And robust expansion is very critical in the sense that by generating higher number of doses, you could treat several patients, which in turn reduces the manufacturing cost, granting higher commercial potential for the product. Next slide, please. So the cells were frozen down on day 21 and then were thawed later to characterize the functional profile of ALA-101 by testing the ability of these cells to kill the tumor cells. Since we wanted to test the activity of the engineered CD19 CAR, we used a tumor cell line model, the SCM cell line. And as you can see from the first FACS plot, the SCM cells express CD19, but not CD1D. The SCM cells, originally the cells originated from a patient with an aggressive B-Cell Acute Lymphoblastic Leukemia, or in short called BALL. The middle figure shows the extensive killing of the SCM tumor cell line by ALA-101. Here, the y-axis illustrates a percentage of cytotoxicity or percentages of tumor cells killed by ALA-101 at different effector to tumor cell ratios, which is represented in the x-axis. Then we tested the ability of the ALA-101 to eliminate the CD19 positive tumor cells from a biopsy sample of a marginal zone lymphoma patient, which is on the extreme right. You could see that the ALA-101 eliminated almost all CD19 positive tumor cells compared to that of the non-engineered iNKT cells, meaning that these cells are capable of killing tumor cell lines as well as primary patient tumor samples. Next slide, please. As Michael mentioned before, ALA-101 is dual targeting when you engineer with a CAR. In the previous slide, I showed the activity of the ALA-101 through the CD19 CAR. And the data presented in this slide shows the activity of ALA-101 through the ITCR that is naturally present in these cells. The ITCR recognizes glycolipids presented on the CD1D molecule of tumor cells. And for demonstrating the activity through the ITCR, we utilized a cell line that overexpressed CD1D, which is called C1R CD1D. And these cells also express CD19. And the graph on the left side shows the killing of tumor cells without the ligand for CD1D. And the one on the right side shows the potentiation of the ALA-101 killing activity with the addition of a non-ligand of CD1D. And the implication is that ALA-101 is more efficient at killing tumor cells when both CD19 and CD1D are present on the tumor of the patient. Next slide, please. Once we found that the cells were effective in lysing the tumor cells in vitro, we wanted to investigate the in vivo activity of the frozen ALA-101 through the CD19 CAR. And specifically, we used the SCM cell line that I described before for this in vivo study. Just a recap that this tumor cell line is derived from a patient with aggressive B-cell leukemia. And these cells express CD1D, but not express CD19, but not CD1D. So specifically, the activity you are going to see is through the CD19 CAR. And these tumor cells also express a luciferase gene that would enable us to monitor the tumor progression in mice by live imaging. So here I'm showing the experimental design employed for the study. To establish the tumor in the mice, similar to that of in the human, the tumor cells were injected intravenously into the mice. Once the tumor was established, as you can see, as fluorescence in the mice pictures shown below. This is the images on day -1 prior to the treatment. And then the animals were randomized and grouped on day -1. And there were 5 mice assigned for each treatment. Then on day 0, the mice were treated with either vehicle or 5 million cells each of non-engineered iNKT cells or ALA-101 intravenously. And then the animals were monitored for tumor growth or survival for a period of 40 days after the treatment with ALA-101. Next slide, please. So as you can see from the graph on the left side, a significant regression of the tumor was observed in the group treated with ALA-101, which is the green line in the graph, as indicated by the reduced fluorescence compared to the other group, displaying the anti-tumor activity of ALA-101 just in 8 days of infusion. Illustration on the right side shows an example where in a mouse treated with ALA-101, the one on the extreme right, showing the reduction of the tumor burden indicated by lack of fluorescence compared to the other two mice after 8 days of treatment with ALA-101. And I believe this is an amazing data in an in vivo model with the aggressive B-cell leukemia. Next slide, please. And these mice were further monitored for 40 days after the treatment to see that, whether the ALA-101 provides better survival rate in these treated animals. And as you see in the figure, indeed, ALA-101 significantly increased the survival of these mice compared to that of the two control groups. And this indicates that in vitro response that observed before can be translated in an in vivo tumor setting. And taken together, we presume that ALA-101 has the potential to be effective off-the-shelf therapy for CD19 positive cancers in the clinic. Next slide, please. So, as I mentioned before, the cells were frozen down after the expansion phase. So here we tested the ability of ALA-101 to multiply in response to the CD19 antigen. And for this purpose, the cells were labeled with a fluorescent dye so that we can track the proliferation of ALA-101 based on the dilution of the dye. The labeled cells were then exposed to fresh tumor cells that are either positive or negative for CD19 for 3 consecutive days, and the proliferation of the cells were monitored after the 7 days of the first stimulation. And it was monitored in the presence or absence of IL-15, a cytokine that supports the proliferation of immune cells in humans. As you see in the picture, the green histogram indicates the proliferation of ALA-101 when stimulated with CD19 positive tumor cells. The more the proliferation, the histogram shifts towards the left side as the cells tend to lose the labeled dye after several divisions. A pronounced proliferation of ALA-101 was noted in the presence of IL-15, and also it is worth to note that the cells also proliferated in the absence of any exogenous IL-15 when stimulated with CD19 positive tumor cells. And as we know, each patient is different from another one, even though they have similar forms of cancer. And this is an exciting and very important data set that provides an insight on how ALA-101 behave in patients upon continued exposure to cancer cells in the presence of absence of cytokines in patients, thereby enhancing the persistence and in turn efficacy of the ALA-101. Next slide, please. So our next test was to test their ability to secrete cytokines when stimulated with CD19 positive tumor cells. And here we are showing the upregulation of a few cytokines, like examples, granzyme B, TNF-alpha, and perforin, as indicated by the green in their respective histograms. And many more that we don't show here actually shows that, the ability to secrete cytokine potentiate the efficacy of ALA-101 in addition to the direct cytotoxic activity. The next slide, please. To summarize, Arovella's proprietary manufacturing process allows efficient expansion of iNKT cells without compromising their functional attributes. Expansion of these cells are critical for manufacturing to produce multiple doses from a single run in order to address the manufacturing cost and logistical challenges of current autologous therapies. And we generated the ALA-1 using a 3rd generation antiviral vector with a proven safety profile from Lentigen Technologies in preparation for the manufacturing of the clinical material for our trial. And ALA-101 conferred substantial anti-tumor effect and significantly extended the lifespan of mice with an aggressive B-cell acute lymphoblastic leukemia. And so what it's like, taken together, we presume that ALA-101 has the potential to be enough effective off-the-shelf therapy for CD19 positive leukemias and lymphomas in the clinic. And Arovella continues to progress the ALA-101 towards first-in-human trials. And with that, I hand over to Michael for the rest of the presentation. Thank you.
Great, thanks, Mini, and thanks very much for taking us through the data as you described. Right, it does give us the confidence of really rapidly accelerating and working towards taking this product further through the manufacturing stages and into clinic. And I would recommend for anybody that's interested in viewing the poster itself, it is available on our website. If you have a PDF of these slides, you should be able to access it by clicking on this poster picture itself. But of course, if not, please just do go to our website and you should be able to find it or locate it there. And while we're at the conference, it was wonderful. We had a 4-hour poster session, and this was a good representation of the level of activity and engagement of people coming around to discuss the data and the technology with us. So we had a number of great discussions, of course, some that we'll continue to follow up. But I think for Mini, certainly wasn't a 4-hour period where she had a lot of time to rest. Certainly a lot of talking was done, which is good. So in terms of where we are at the moment, this is a key focus for us, clearly, to take ALA-101 through the next steps and to get the manufacturing completed and then take this product into clinical trials. So already what we have done is optimize the viral vector to engineer the CAR with regulatory friendly elements and working with the 3rd generation lentiviral system. So we're pleased that that's now done. It is a key step in terms of the manufacturing. Now, also, given that we are working with an allogeneic product, we've worked hard to identify high frequency iNKT T-cell donors, and we'll continue to have that as part of our workflow because they do become a key raw material. And throughout the remainder of H1 2023, we'll continue to optimize the manufacturing process to produce clinical grade material. And of course, through the second half, we intend to scale that up and then start generating the data that's required to submit to regulators and ultimately to produce clinical trial material and take that into phase 1 clinical trials. Now, just shifting gears a little bit, I think I'd like to just highlight some of the other things going on in our platform and pipeline. So we've talked through ALA-101 already. That's what we're rapidly advancing towards clinic for CD19 producing cancers like lymphoma. Of course, with the partnership with Imugene, it's quite exciting because we can take the OnCARlytics platform and the CD19 CAR platform and combine them. The oncolytic virus will essentially target and tag various solid tumors with CD19, which then opens up the potential for us to target really any cancers that the oncolytic virus is able to infect and label them with CD19. And of course, our DKK1 peptide targeting CAR, we've data to suggest that's beneficial for not just multiple myeloma, but also a range of solid tumors. Now, I just want to highlight that we do have, it is a platform in the sense that we can start to work with additional chimeric antigen receptors that we either choose to create or get our hands on. And this could be across both blood cancers or solid tumors. We can consider things like having 2 chimeric antigen receptors added into the iNKT cells. So then for cancers that have CD1D on the surface, we actually have a triple targeting product. Now, over the next 6 to 18 months, let's say we've got a lot planned, and it's going to be a very busy time for the company. But as I've just reiterated, I think the key point for us is really working through the manufacturing of ALA-101 and then taking that into phase 1 for patients that have CD9 producing cancers like non-Hodgkin's lymphoma. And so for the Imugene partnership, we're looking to generate additional proof of concept work. We've already generated some promising in vitro data, which has now enabled us to make the decision to move forward into the animal studies, which is great. And then once that's completed and we're happy with those studies, we'll move into the more formal IND enabling work for the combination of the 2 drug platforms. And for ALA-104, we're continuing to validate that product and work towards then taking that and combining it with the iNKT cells. So I would just want to reiterate, the cell therapy space is something that has revolutionized the way we think about cancer treatment. At one point, it was an emerging field. Now it's well established, but certainly what we see for the T-cell space, and this is a snapshot of companies in that space, there is quite a lot of activity. It is quite busy. And we do see the same for the NK cell space as well, where there are quite a lot of players already in those areas, whereas still for the iNKT cells, this is the majority, or really, I think all the companies we found of significance that are working on CAR iNKT cells for cancer treatment, it's a handful. So we're excited that we're in a niche area of what is really the cutting edge field for oncology or cancer treatment. It's also pleasing that despite the headwinds that have hit the sector over the last, say, 18 to 24 months, there are still a number of transactions transpiring in the cell therapy space. And of course, for us, it's pleasing that a range of them are also in the preclinical stages, and then, of course, into phase 1 and phase 2. But even in the preclinical stage transactions, we're seeing that the upfronts are still quite substantial, and associated milestones are also quite attractive. So again, promising and great that we're still seeing activity across the sector. And that just brings me to the financial slide for Arovella. I'd just like to provide an overview. And as I just mentioned, we have seen a number of preclinical transactions, and it's despite, I think we've had a great start to the year through share price appreciation for Arovella, we're still valued at 60 million, which is in some ways below even just the upfront payments for some of those transactions that we've seen recently. So I think with the platform that we're developing and the products that we've got underway, I think we do see that there, and with the team in addition to that, we do see that there is certainly room for appreciation and growth in the company. As at the end of December last year, we finished with 5.2 million cash in the bank, which includes the proceeds from a $1.65 million placement that we completed in January of this year. Now, in terms of our shareholders, we're very pleased with the level of support that we've received from Merchant Group. They now have over 10%, which is great, and pleased with their support. And we also have a number of supportive high net worths and small institutions on the register, but of course, always looking to have those that understand the risks that come, of course, with the biotechnology sector, but of course, the potential returns that can come in the space. So certainly looking to welcome additional members onto the register. And I'd just like to finish with, again, a snapshot of the team, because it has grown and it continues to grow with an absolutely stellar group of people. I'm delighted that we had Tom join as the chair of the board only recently, but I think what we've got across, really, it is still quite a small group, is an enormous amount of horsepower and experience across not just the one stage of drug development, right the way through from research and development through drug development itself, and then into commercialization and the transactional experience, which is great. And it'd be remiss of me not to reference and just to mention our thanks to our colleagues at Imperial College London, who have contributed to some of that the experimental work that we presented at Aacr at the conference recently. And just with that, I'd just like to say, again, a big thank you to everybody for taking the time to listen in today. We think it's great data, and we're very excited to continue accelerating through the manufacturing stages and into clinical trials. And I'd just like to highlight, if for those of you that would like to learn more and didn't get a chance to attend our explanatory webinar, it's a great overview of the iNKT cells in a little bit more detail, and certainly provides a lot more context. But if you're interested in that, certainly click on the picture or go to this link down here below to get a chance to listen to that. And again, I'd just like to thank everybody for their time, and we'd be delighted to take any questions.
Thanks, Michael, and to Mini as well. We'll move on to the questions as you've mentioned. Again, if anyone has one that they want to submit live, please feel free to do so using the Q&A option or panel within Zoom. The first question is, what is the relevance of proliferation of ALA 101 in the absence of IL-15?
Maybe I can take that question.
Great.
So in general, when you do allogeneic CAR T cell therapies, the patients will be lymphodepleted prior to giving the cells. So one intention of the lymphodepletion is actually to remove all the cells that use all the cytokines in the patient system to be utilized for the infused cells to grow. And so, as you know that, different patients behave differently, and they do have different levels of cytokines in the system. And sometimes they have very low, sometimes really have very high levels of IL-15 and other cytokines in the system. And IL-15, one of the important one considered as a homeostatic cytokine that drives the proliferation. So what you can see from this, the cells continue to proliferate after 7 days after seeing the tumor. Actually, it is indicated that, even with a low dose or less because you can see supraphysiological doses, they proliferate, but even without cytokine, they proliferate, but there will be a little bit of cytokine available in the patients, actually will promote these cells to proliferate further and actually continue to expand, and which will improve the persistence of the cell, thereby killing the tumor cells in vivo. So actually, it's a pretty important data set that we generated, and it's very important because you can see that it is very well documented in the CAR T cell space, that in the importance of IL-15 and those IL-2 mediated proliferation in the patients.
Great, thank you. The next question is, what does this data mean for the progress of ALA 101?
I'll take that, Matt. So it's, as I said, we licensed this technology from Imperial College London some time ago, and over that time, what we've managed to do is take that original concept and start making it, into a product essentially, so that we've got components that will meet the regulators' standards. And what we've been able to do since that time is to go on to demonstrate really excellent data with the product as it stands today, which is ALA 101. So it is significant for the company. And now, of course, the key steps are for us to accelerate through manufacturing and take it into clinic. But yeah, it's really exciting for the company, obviously, and great that we've managed to do that and get the data that we were hoping to get.
Thank you. The next question is, can you tell the viewers of timeframe for clinical trials and ultimate production?
Yeah, so that's something we're working through at the moment. Key steps are, as I said, we've created the lentivirus, that's probably the first and most important component. So that's done. And now what we're working through is the optimization of the process itself and the scale up. So I won't give a time frame exactly. That's a difficult thing to do right now. But of course, as we move through that process, then we get more data that supports when that will take place. And then once that's done, we can start submitting to regulators to file for phase 1. So look, we've said that second half of the year we'll be looking to scale up, and I think that's where we're aiming or intending to be. So hopefully it's not too far away. Now, in terms of just making it clear, so for production, if that was the second part of the question, we don't expect to be making commercial product for some time. We still need to go through phase 1 clinical trials, phase 2 clinical trials, and so forth before the product gets approved. Just making that clear.
Thanks, Michael. I'll just get you ready to go to some certain slides here, as there's references to a few of them. So on slide 10, the question is, what does the middle graph on slide 10 indicate? And what does toxicity remaining at 15 to 20% mean for safety of patients?
We'll get there.
You want me to take the question?
Yeah, if you've got the slide handy, Mini, go for it.
Yeah. So the slide that is showing, the slide shows the cytotoxicity of ALA 101 to SEM cells in vitro. That is a 24-hour assay period. It's an in vitro assay, and we actually stopped those co-culture after 24 hours. So if you continue to keep the cultures for longer period of time, and you can see that all of the cells get killed because they also secrete cytokines, which also kill the tumor cells. And so we believe that this is an in vitro data. So if in vivo, they continue to proliferate, and then they will kill the remaining cancers. As we have shown in the previous webinar, we have shown a model where, completely eliminates the tumor, that is the continued persistence of the cells and activity of cells. So yeah, and that is what it tells you. And it's an aggressive model as well.
Thank you. Now referring to slide 14, the question is, how does one read the graph on slide 14? Does it show that the benefit of ALA 101 is 5 more days of life only, and can it be improved?
Okay. So this is our first study, and this is the first batch of ALA-1 created with the new lentivirus that actually we are preparing for our clinical trial. So we used a small dose of iNKT cells, and we haven't done any dose titration studies in a particular model of this. And this is a very aggressive model, and anything that is 5 days is a significant benefit in the patients as well. And these cells grow really fast. I mean, we started with 500,000 cells, and you know the aggressiveness of the model where the tumor takes up really fast and kills everything. So, and we also wanted to dissect the functional ability of the CAR. That's why we use smaller number of cell, dose in this particular study. But we will be running dose titration studies, and most of the CAR T cell studies need even 10 million cells in the mouse to get rid of some more aggressive models like this one. So we will be running more dose titration studies, which we will be able to show that elimination of these cancer cells in the future.
And Mini, probably worth pointing out, this is a cancer cell line that also doesn't produce CD1D.
CD1D.
We made this as hard as possible for our product, and again, to see the data like this is really encouraging and exciting.
Right.
Thank you. Now going back onto slide 13, Michael. The graph on slide 13 shows the ALA treatment with increasing radiance, albeit less than controls, but the pics show the cancer disappearing in the mice. Can you please explain?
I'm happy to take it, Mini, and then you can jump in and add anything
Sure.
else. Essentially, what we're looking at here on the graph, it's correct. It's an increase in radiance over time, and what we're looking at in the animal pictures is it's the signal inside of the animals. So based on the scale, that's how strong the intensity will be. And so when we think about here in the animals, we're actually seeing that it's not a reduction, but we've got 90% less or whatever percent less in the particular control animals here. So it's because of a scaling issue that we see it looks like it's disappearing, but it's just that there's far less that's able to propagate inside the iNKT cell treated animals.
And in order to add to your point, Michael, actually, this is the activity showing at day 8. And, since this is an aggressive model, they continue to grow, they proliferate, it's aggressive, so, that's why the radiants keep increasing. But you see that the difference from the control, they continue to check on the, tumor. So the difference between the controls is being attributed to the early activity of the iNKT cells when there is enough effector to target ratio was there. So that's why the radiants keep increasing.
Thank you. And this will be the final one unless anything else comes in. Can Michael comment on whether any other iNKT cell companies presented at AACR, and how does the data compare to others in that space, in your opinion?
Yeah, good question. And there was only one other company that presented related to iNKT cell therapies, which was MiNK Therapeutics. So we know them quite well already. And, they are pioneering some things in the field. What they work with is, so imagine we've got the iNKT cells alone without a chimeric antigen receptor. That's their lead product in clinic, and that was what they presented their poster on. And that gives us a lot of important information about the safety profile of iNKT cells. So in that experiment, they put up to 1 billion cells into humans, plus and minus PD-1, and saw some level of activity in those patients. But again, that's when they're not engineered with a CAR or the targeted missile to go and recognize and find and kill the cancer types. And it was also in solid tumors, which is a higher bar. But certainly from our perspective, I believe there was no cytokine release syndrome, no neurotoxicity in the brain. So it's really exciting data from that perspective, and gives us confidence to accelerate moving forward. So in terms of the data and how our data compares here, it's a little bit difficult to compare because it's not apples to apples, given that we're specifically looking at CAR iNKT cells, and we will not be working with engineered. But certainly, again, right, MiNK's a $70 million company in the US, and phase 1, but preclinical for their CAR assets. So it's exciting for us as well, and looking forward to getting our products into clinic.
Great. Thanks again, Michael and Mini. Michael, I'll throw it back to you just to provide a closing comment.
Great, thanks, Matt. And I'd just like to finalise by saying, actually, excellent questions. Thank you to everybody for taking the time to listen in and provide those questions. And we're certainly looking forward to giving more updates throughout 2023 as we progress through the manufacturing stages. Thank you.